This is a video to help you through the activity, Components of Nucleotides. The figure on this activity shows that nucleic acids, which we know as DNA and RNA, can be broken down into simpler units through hydrolysis reactions. These simpler units are called nucleotides. The nucleotides are what we will focus on in this activity. Nucleotides can be broken down into simpler units through hydrolysis again. The simple units that make up a nucleotide are nitrogenous bases, sugars, and phosphate groups. Nitrogenous bases, sugars, and phosphate groups come together through condensation reactions to form nucleotides. And nucleotides come together to form long polymers called nucleic acids. This is what we know as DNA and RNA. If you haven't already, please take a moment now to look over the first 2 pages of this activity. The information you need to answer these questions is written for you in the activity. Please read carefully and try to answer the questions now. The basic building blocks of nucleotides are nitrogenous bases, sugars, and phosphate groups. In this activity, we're going to focus on the condensation of these components to form nucleotides and their hydrolysis into the basic components. Let's take a closer look at these simple components. The nitrogenous base is the nitrogen-containing aromatic compound that is attached to the sugar in a nucleotide. The nitrogenous bases that make up DNA and RNA are shown in front of you. There are 2 types of nitrogenous bases, purines and pyrimidines. The general structure of a purine and pyrimidine are shown in the dotted boxes, and their numbering system is also shown. From this numbering system, it's important to be able to recognize where N9 is in a purine and where N1 is in a pyrimidine. A pyrimidine will always be connected to a sugar at N1, and a purine will always be connected to a sugar at N9. The purines that are part of RNA and DNA are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. Let's look at the sugar part. There are 2 possibilities for the sugars, ribose and deoxyribose. The prime on the numbers of the sugars is used to differentiate the numbering system from that of the base. So if I tell you that I'm talking about 2', you know that I'm talking about the carbon on the sugar rather than the carbon on the base. Ribose is the sugar that is found in RNA. RNA stands for ribonucleic acid. Deoxyribose is the sugar that's found in DNA. DNA stands for deoxyribonucleic acid. The difference between a ribose and a deoxyribose is that deoxyribose has been deoxygenated at position 2'. Notice that ribose has an alcohol functional group on carbon 2', and deoxyribose does not have that alcohol functional group. Deoxyribose is the sugar in DNA, and DNA stands for deoxyribonucleic acid. So this is the sugar that you will find in DNA. There are 4 bases that are present in DNA. Adenine, guanine, cytosine, and thymine are present in DNA, but uracil is not. Rather, uracil takes the place of thymine in RNA. RNA stands for ribonucleic acid, and ribose is the sugar that's found in RNA. There are 4 bases that make up RNA. They are adenine, guanine, cytosine, and uracil. Adenine, guanine, and cytosine appear in both DNA and RNA. Thymine is only found in DNA, and uracil is only found in RNA. Uracil is the analog to thymine in RNA, and thymine is the analog to uracil in DNA. Take a moment now to make sure you understand these basic components before we move on to phosphate. The sugar attaches the nitrogenous base to the phosphate. Let's take a closer look at the phosphate. As a review, try to draw the Lewis structure of phosphate. It's helpful to start with the Lewis dot diagram of each atom. The formula for phosphate is HPO4 2-. I have 1 phosphorus, which has 5 electrons, 4 oxygens that have 6 electrons each, 1 hydrogen with 1 electron, and then I have 2 extra electrons from the 2- charge on phosphate. Phosphorus is an atom that breaks our normal bonding schemes, and it makes 5 bonds. You would expect it to make 3 bonds like nitrogen, but phosphorus is special and it actually can make 5 bonds and have a neutral charge. To draw a correct Lewis structure, always put the atom that makes the most bonds in the center, and then try adding other atoms that make multiple bonds first. In this way, I would put phosphorus in the middle, and I would surround the phosphorus with oxygen. To make sure that all of the oxygens have correct bonding schemes, I would make a double bond between oxygen and phosphorus. After you surround the phosphorus with oxygen and try to make as many correct bonding schemes as possible, then you can add hydrogens and extra electrons. This is the Lewis structure for phosphate. Now let's look at what happens when phosphate, sugar, and bases come together. This part of the activity tells you specifically how each group is attached. It tells you that the phosphate's hydroxide group will condense with the alcohol group of the C5' of sugar. Notice the notation that's being used here. The C5', even if I didn't tell you it was of the sugar, you would know that it's part of the sugar because of the prime. So what this is telling you is that the alcohol group of a phosphate combines with the hydroxide group of C5 of the sugar. I'm going to use ribose as my sugar here. Here's C1, 2, 3, 4, and 5', and this is the alcohol group on C5'. In this case, I'm going to remove the O from the atom that has the most bonds to oxygen, so the phosphorus. I'll remove this alcohol functional group, and I'll remove the hydrogen of the other alcohol. And I'll make water. A new bond will form between the oxygen of C5' and the phosphorus. This is what it would look like. Here I've made a new bond between the oxygen of C5' and the phosphorus. Water is a product because this is a condensation reaction. This is not a complete nucleotide yet. The next part of the problem tells you that a purine's N9 will condense with the OH on C1. In number 7, they're asking you to draw the nucleotide with adenine, which is adenosine monophosphate. The condensation reaction happens between the alcohol group on C1. C1 is the anomeric carbon of the sugar. And it happens with N9 of adenine. Here's adenine and here's nitrogen 9. I've labeled it here. And the hydrogen on this nitrogen will be removed and combined with the alcohol on the anomeric carbon to form water, and a new bond between the anomeric carbon and N9. Here I've labeled N9 again for you. Notice that I make an additional water molecule through this condensation reaction. There are 2 new bonds that are formed in the reaction shown. The new bond between the phosphorus and the oxygen is called a phosphoester bond, or a phosphate ester bond. This functional group is a phosphate ester. It's similar to a regular ester, except instead of a carbonyl bound to an oxygen, it's a phosphate bound to an oxygen. The other new bond is called an N-glycosidic bond. Just like a bond between 2 monosaccharides, the bond between a monosaccharide and a nitrogenous base is called a glycosidic bond. In this case, it's called an N-glycosidic bond because the anomeric carbon is bound to a nitrogen. This new molecule is a nucleotide, and it's called adenosine monophosphate. Let's talk about naming. This is the same molecule that I just showed you. It's adenosine monophosphate. When you combine a phosphate plus a sugar plus a base, this type of molecule is called a nucleotide. And this one is named adenosine because it has an adenine base, and monophosphate because it has 1 phosphate group. It is abbreviated AMP for adenosine monophosphate. A simpler version of this is called a nucleoside. A nucleoside is the sugar plus the base without the phosphate group, and this nucleoside is called adenosine. You take the name of the nucleoside and combine it with the number of phosphate groups to make the nucleotide name. The base in this case is adenine. The base name is different from the nucleoside name, and that is different again from the nucleotide name. There's a table in your textbook that lists the name of all of these things. Here I've put the structure again for you. This is table 11.1 in your textbook. The bases in RNA are adenine, guanine, cytosine, and uracil. And the bases in DNA are almost the same, except instead of uracil, you have thymine. The base is this part. The structure I've shown you here is adenine attached to a sugar and a phosphate. So this is adenine, but you could also replace this with guanine, cytosine, uracil, or thymine. When you add the sugar, then you have a nucleoside. When the sugar is ribose, you simply change the ending. Here we're talking about the nucleoside, or what I have in the blue box on this figure. The nucleoside of adenine is called adenosine. Guanine is called guanosine, cytosine is called cytidine, and uracil is called uridine. If instead you use deoxyribose, you simply put deoxy in front of the name. Adenine with deoxyribose is deoxyadenosine. Guanine with deoxyribose is deoxyguanosine, and so on. When you add a phosphate group, if it's just one phosphate group, you simply add monophosphate to the end of the name of the nucleoside. Take a moment to examine this table and make sure that it makes sense to you. The nucleotide is phosphate sugar plus base. The nucleoside is just the sugar in the base, and the base is simply the nitrogenous base, the purine or pyrimidine, from the first page of your activity. Let's talk about the difference between the structure of something like AMP, which is adenosine monophosphate, and ATP, which is adenosine triphosphate. The difference in the structure is simply that the triphosphate has 3 phosphates, or it has 2 more phosphates than the adenosine monophosphate. In the name AMP, the M means mono for 1 phosphate, and in ATP, the T means tri for 3 phosphates. Adenosine monophosphate is one of the basic building blocks of DNA, and ATP has almost the same structure, but this is the energy molecule of your cells. These types of molecules are used for many different purposes, from DNA to energy. Notice that I've drawn both of these structures with the ribose sugar. If I wanted you to draw this with a deoxyribose, I could indicate that using the name deoxyadenosine rather than adenosine, or I could show you with the abbreviation DAMP instead of AMP. Notice that the difference between these two structures is only the hydroxyl group at the C2' of ribose has been removed in the deoxyribose. And here's the structure of deoxy-ATP or DATP. Notice that the D indicates that this is a deoxyribose sugar as part of the nucleotide. Thanks for watching this video on components of nucleotides. Please notice that we've only talked about this part of the equation. In the next activity, we'll talk about the formation of nucleic acids like DNA or RNA from nucleotides. And we'll also talk about the structure and properties of nucleic acids. If you have any questions about this video, please feel free to post them in the comment section. And if this video was helpful to you, please let me know by giving me a vote.